Then hello, good morning, everybody. I am Dr. Walvekar. In the continuation of metabolism of the carbohydrates, in the last class, we had discussed about the glycolysis. Glycolysis is a major pathway in the carbohydrate metabolism, and it is a path, pathway wherein the fuel in the form of ATP is produced once the glucose molecule gets converted into finally the pyruvate. So at the end of the reaction, it gives about 2 molecules of ATP, pyruvate, NADH in this one. This is the pathway which occurs in both aerobic and anaerobic situation. So it is called as the aerobic and anaerobic glycolysis pathway. So it is the pathway which involves about the 10 steps wherein it will be coming across the glucose molecule, coming across the various enzymes, and then the products will be formed. What the products that will be formed will be the starting material for the next reaction. In that way, but at the end of the 10 reactions, the pyruvate will be formed in the aerobic reaction, whereas the lactate will be formed in the anaerobic reaction. Almost all of them are the reversible reactions, except at the 3 positions, it is the one which consists of the irreversible reactions. So irreversible reactions are there to regulate the pathway or to check the formation of ATP. So today we will concentrate on the regulation of glycolysis at 3 positions where the enzymes involved like the hexokinase, phosphofructokinase and pyruvate kinase. So we will see that one, how this one takes place. So we are seeing at one glance how this glucose is getting converted into the pyruvate. So first phase, it is divided into the 3 phases. One as the initial phase or investment phase, where there will be the utilization or the use of the ATP will be there, and then at the place where the fructose 1,6-phosphate is converted into the glyceraldehyde phosphate hydrogenase, that will be referred as the splitting phase, and then finally at the 7th one it will be called as the production phase or beneficial one, you can call it. So the involvement of the enzymes are hexokinase, phosphoglucose isomerase, phosphofructose kinase, aldolase. Like that, all the enzymes are there. In this one, at the 3 places, it is irreversible. So that is the one which regulates this mechanism or the pathway. So glycolytic pathway has got the 2 roles to play. That there, one is the generation of ATP and providing some precursors for other things like the fatty acids or the formation of the fatty acids for the lipid purposes. And the enzymes which are carrying out these reactions, they themselves act as the regulating parts. So these irreversible reactions, they are also referred as the rate limiting steps because they are serving as the regulating valves so that they can operate in an open and closing way. That means whenever there is the formation or enough amount of the glucose or in the form of ATP is there, they can stop the pathway, and then when there is the deficiency of that one, the ATP, they can start that one. In that way, they maintain or they regulate the path of the glycolysis. So, in the glycolysis, all the reactions, as I said, they are reversible, except the reactions which are catalyzed by the hexokinase or the glucokinase, next the phosphofructokinase and pyruvate kinase. So, these reactions of these three reactions of the glycolysis, they are one which act as the reversible one. And these are catalyzed by the allosteric enzymes. Allosteric enzymes means an enzyme will be having some active site. All the enzymes will be having the active sites. Apart from the active site, if the attachment or binding of the effector molecule is taking place at other place than the active site, then it is referred as the allosteric enzymes. Now, what is the importance of this regulation? The main important function of glycolysis is the, you know well, that is the production of the energy in the form of the ATP, and situation wherein there will be the requirement of ATP, then only it should be generated. That is the main hidden agenda or the agenda of this regulation. So, as the glycolytic intermediates feed into the several other pathways, the regulation of the glycolysis, it occurs more than one point. In this one, at the three places it gets regulated. Similarly, in other pathways also, the regulations will be there. Those will be discussed as and when we come across those pathways. For example, the activity of an enzyme like phosphofructokinase has to be regulated because it is the one which is considered as an important pathway in this or important step in this pathway. And also it consumes the ATP, which is a valuable resource for the cell. It means there should not be the waste of the ATP also. That way, all the reactions are regulated and they are coordinated. So, in one step, the dihydroxyacetone phosphate, it is the precursor for the glycerol component of the lipids. It means if this can be managed or maintained, then only the availability of the product that reforms dihydroxyacetone phosphate, that will act as the precursor for the formation of glycerol components of the lipids. Hence, the regulation of this pathway is essential. So now coming to the first two enzymes, they are actually, though they are referring two examples, two enzymes, they are acting upon only the one part only. That is, you know well, glucose is converted to glucose 6 phosphate, and glucose 6 phosphate cannot get converted into the glucose. It is irreversible one because whatever the product that is formed, glucose 6 phosphate, it will be impermeable for the membrane of the cell. So there, the enzyme that is acted upon is the hexokinase. This is an allosteric enzyme. Just in the previous slide, we have seen that one. What do you mean by the allosteric enzyme? So what happens? The action of hexokinase will be regulated or inhibited by the product itself only. When there is enough amount of the glucose 6 phosphate is converted into the glucose 6 phosphate, which is impermeable by the cell membrane, then that one acts as a regulation for that enzyme, that is hexokinase. So this is the reaction where the phosphorylation will be taking place in the presence of magnesium as the cofactor. Hexokinase act upon the glucose and get it converted into glucose 6 phosphate. Now, so there is a presence of the glucokinase also. You may be remembering that the presence of hexokinase is in the, or it is found in the, all the other, almost all the cells except the liver and beta cells of the pancreas. Whereas the glucokinase is present in the liver and beta cells of the pancreas. The thing is that the glucokinase is not that much having the affinity for the glucose. So what happens when there is more amount of the glucose formation or entry of the glucose and getting converted to glucose 6 phosphate, then only the glucokinase act upon. Even if it is lesser amount of the glucose is formed or converted into the pathway, there the hexokinase comes into the picture. So the slide wherein the graph shows that one, the glucokinase, which is present in the liver and beta cells of the pancreas, they get acted upon the glucose when the glucose molecule has got the value or the glucose value is more than 100 milligram per deciliter. Whereas hexokinase is the one which is getting acted upon on the glucose only when even if the low amount of the glucose is present there. In this graph, glucose is expressed not in the milligram per deciliter, but it is millimole per liter. So 18 milligram gets to the equivalent to 1 millimolar of the glucose. So glucokinase as such, it is the isoenzyme of the hexokinase. So it is highly specific for the glucose and it is expressed only in the liver and pancreatic cells. And another thing to be pointed out here is that this glucokinase will not be inhibited or it will not be regulated by the glucose 6 phosphate. Whereas the glucose 6 phosphate, what is the product that is formed, it is inhibiting the hexokinase. It should be noted here. And another thing, when once the action of this glucokinase takes place, it takes place only when the high amount of the glucose is in the reaction. So that time it gives the signal to the insulin to get released, synthesized, and then the action of insulin will take place there. So the release of insulin takes place and the pathway of glycolysis will be regulated. So in the first part, you should understand that the regulation is taking place at the first step of the reaction, that is glucose getting converted into glucose 6 phosphate. Glucose 6 phosphate itself, the product becomes or it is inhibiting the hexokinase, whereas it is not inhibiting the glucose 6 phosphate, it is not inhibiting the glucokinase. And if the more amount of this glucose is there, then there will be the release of insulin and the glucose level will be regulated. Now coming to the second one, that is phosphofructokinase, PFK. So in the continuation of these reactions, you must have seen that one fructose 6 phosphate, it gets converted into fructose 1,6 bis phosphate in the presence of phosphofructokinase 1, that is the enzyme. And there what happens, there will be the conversion of ATP into the ADP. This is the another phosphorylation takes place, that is at the step where fructose 6 phosphate is converted into fructose 1,6 phosphate. Now just we go a little bit elaborately. It is an important regulatory enzyme and it is the one which is involved in the feedback of the control. ATP is used as the ATP, the phosphate donor. So here, I just move to the next slide and come back here. You can see that one, this reaction, fructose 6-phosphate get converted into fructose 1,6-bisphosphate. It is the direct reaction that takes place in the presence of phosphofructokinase 1. But what happens where the regulation takes place? When there is more amount of the ATP and the citrate that is formed from the citrate synthesis. So these are the two things which will inhibit the formation of phosphofructokinase. Whereas the presence of AMP and fructose 2,6-bisphosphate, they will act as a activators. The fructose 2,6-bisphosphate is the another isoenzyme of the phosphofructokinase, and this will react will show in the next slide. So now I come to again this one here. ATP and citrate, they are the inhibitors. In this reaction, the reaction where the fructose 6-phosphate is converted into fructose 1,6-bisphosphate, the two things will be there, that is ATP and citrate, they are acting as the inhibitors. AMP and fructose 2,6-bisphosphate, they are acting as the activators. Okay. Now, AMP, it is the one which is promoting the glycolysis. It acts as the indicator of the energy status of the cell. So usually what happens, ATP concentration will be higher than the AMP. But what happens, if there is a small decrease in the ATP concentration, there will be severe rise in the level of the AMP concentration. This mechanism allows the activity of the fructokinase to be highly sensitive for even slight changes in the energy status of the cells. Hence, it can control the glucose that would undergo glycolysis prior to the entry of the acetyl-CoA in the TCA cycle. So, the regulation in this one will be taken by the two things. One is ATP, citrate. These are the one which acts as the inhibitors, whereas the AMP and 2,6-bisphosphate, they act as the promoters or the activators. Now, how the reaction will take place, we will see. You have seen that one, fructose bisphosphate and AMP are the activators. Now, you see that one, first fructose 6-phosphate, it is not getting directly acted upon by the PFK1, but it will be acted upon by our fructose 6-phosphate is having the bivariant reaction, or the actions will be there of in two ways. That is, one, fructose 6-phosphate will get acted upon by the PFK2, that is phosphofructokinase 2 kinase, and it will form fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate, once when it happens, it will split back. It will split back to fructose 6-phosphate in the presence of the one enzyme called fructose kinase, fructophosphokinase 2 phosphatase. Then it goes back to fructose 6-phosphate. From fructose 6-phosphate, it gets directly acted upon by the phosphofructokinase, and then the final product that will be formed is fructose 1,6-bisphosphate. I will explain once again. Actually, this is the irreversible reaction wherein the fructose 6-phosphate, which will get acted upon by the phosphofructokinase 1, it will be getting converted into fructose 1,6-phosphate, bisphosphate. This is the reaction. In this reaction, there will be the involvement of ATP, citrate, fructose 2,6-bisphosphate, and AMP. The regularization will be taken by two things. One is The ATP, the presence of ATP and citrate, if the ATP is in more amount, then it will inhibit the reaction. So it will not allow the process or the reaction to go further into the glycolysis way. Whereas if there is small change or the decrease in the ATP, the AMP will be the one which will activate and it allows the process to continue. This one along with the fructose 2,6 bisphosphate action will be taken place. How this fructose 2,6 bisphosphate takes place formation, that is in this one. Actually the fructose 6 phosphate, it gets converted into fructose 2,6 bisphosphate by the action of phosphate fructokinase 2 kinase. This is the one enzyme which converts fructose 6 phosphate into fructose 2,6 bisphosphate. And this molecule or this product, fructose 2,6 bisphosphate, it will go back or it will split back to fructose 6 phosphate by the another enzyme called as phosphofructokinase 2 phosphatase. By this one, it goes back to fructose 6 phosphate. From this fructose 6 phosphate, the action of PFK1, that is phosphofructokinase, takes place, and the final product, that is fructose 1,6 bisphosphate, will be formed. This is how, by the presence of the AMP, ATP, citrate, and fructose 2,6 phosphate, it will be managed. Okay. So the same thing, it is written here. It acts on, so acts as a stimulator for the PFK1 and also for the further glycolysis. This one also act as a activator, and then this one will act as the activator and it continues the process of the glycolysis. Now coming to the third one, that is pyruvate kinase. The pyruvate kinase is the one enzyme which is involved in the last step, that is the conversion of the phosphophenol pyruvate into the pyruvate, and also there will be the production of 1 ATP, and it is a irreversible reaction. This step, the main aim of this reaction or the step is that it should increase the concentrate of the ATP in the cell, and when once the high amount of the ATP or the requirement of the ATP is formed, then it will inhibit the PK, that is pyruvate kinase. So another thing, these reactions will be also regulated by the hormones also, that is the insulin and also the glucagon. The glucagon increases its activity, whereas the glucagon inhibits. This is the hormonal action of the two hormones, that is insulin and the glucagon. So seen here, phosphophenol pyruvate, it is getting converted into the pyruvate in the presence of pyruvate kinase. And once the enough amount of the ATP is there, it inhibits further formation of the pyruvate. This is how the action will be there by the more amount of the ATP forming, and it inhibits the reaction. So these the three things, or you can consider four things that are in the picture. One is the hexokinase or glucokinase. The second one is the phosphofructokinase, and third one is pyruvate kinase. And last one is that the glycolysis steps or the pathway, it will be regulated by the action of the hormones also. The insulin is the one which increases the rate of glycolysis, and the glucagon is the one which inhibits the reaction. So This is something about the metabolic reaction by the glycolysis and the regulation of the formation of the glucose into the pyruvate, which is regulated at the 3 to 4 places. Now, coming to another one pathway which is related to this, the glycolysis only, that is referred as Leuberin rapport pathway, or it is also called as the Leuberin rapport shunt. This is something a bypass or a supplementary pathway which is operable more in the erythrocytes, where the formation of one compound called as the 2,3-bisphosphoglycerate will be there, which one will help to regulate the oxygen release from the hemoglobin and issuing it to the tissues. This is more beneficial when the persons are in the anemic condition. And second one, when the persons are at the high altitude, wherein they feel short of the oxygen, where this bypass or a supplementary pathway will take place, and its main important aim will be that to make the availability of the oxygen very easily. So this is the supporting or bypassing pathway. So about 15 to 25% of the glucose that get converted to lactate in the erythrocytes, it goes via 2,3-BPG synthesis. So the end product, when it is lactate, definitely you must think that it is the one which is taking place in the anaerobic condition. So here you can see that the pathway is from glucose to pyruvate. What happens when once glycerol 3-phosphate splitting will be taken place, 1,3-bisphosphoglycerate is formed. This 1,3-bisphosphoglycerate, instead of going directly forming the 3-phosphoglycerate, what happens? It gets converted in this condition or in this pathway. It gets converted into 2,3-bisphosphoglycerate. 1,3-bisphosphoglycerate gets converted into 2,3-bisphosphoglycerate in presence of bisphosphoglycerate mutase. This is the one enzyme which which helps or it takes the action or enzymatic catalytic action that converting 1,3-bisphosphoglycerate to the 2,3-bisphosphoglycerate in presence of bisphosphoglycerate mutase. This 2,3-bisphosphoglycerate, then it comes converted into 3-phosphoglycerate in the presence of this enzyme, 2,3-bisphosphoglycerate phosphatase. So it is not coming directly, but through via, or you can say that it is bypassing this pathway and it is getting converted into 2,3-bisphosphoglycerate and then to 3-phosphoglycerate, which further will be converted into the pyruvate like this. This is the supplementary pathway. So why it happens and where it happens? So red blood cells, they utilize the more glucose than their requirement, and it happens because of that one, resulting that there will be the more accumulation of ATP and 1,3-BPC, that is bisphosphoglycerate. So when once there will be the ATP formation will be there, then it ceases the process of the glycolysis. So this pathway, that is Leuberin rapport pathway, it provides the supplementary mechanism for the glycolysis. In which condition we will see that. So in erythrocytes, 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate, instead of converting to 3-phosphoglycerate, it is converted into the intermediate 2,3-bisphosphoglycerate, and 2,3-bisphosphoglycerate is then converted into the 3-phosphoglycerate. This is how the reaction takes place. Now, What is the importance of this intermediate formation of 2,3-bisphosphoglycerate? It is the one which combines with the hemoglobin and it reduces the affinity towards the oxygen. So that what happens in that one, it will help to unload the oxygen more easily in the tissues. The supplementation of oxygen will be done more easy way than other conditions. Particularly under the hypoxic conditions, this concentration increases in the RBC and it helps for the releasing of the oxygen in the tissues even when the PO2 level is low. PO2 means partial pressure of the oxygen in the our breathing or in the blood it is. So the compensatory increase in the 2,3-BPG will be found particularly in the place where there is a high altitude anemia, wherein this process it helps for the oxygen dissociation, and hemoglobin exhibits high oxygen affinity in the exokinase defective patient. So if there is an inherited disease who are suffering from the exokinase defective patients, it has been observed that hemoglobin it exhibits higher affinity for the oxygen. Now we have seen that one by the pathway, the glucose entering into the 10 steps and getting converted into the pyruvate, formation of the pyruvate, and there will be another formation of the ATP molecules. So once the pyruvate is formed, what is the fate of that one? That we have to know. We should know that one. What happens? Apart from the metabolic fate of the glycolysis, it is pyruvate is also formed from the degradation of the amino acid. This one will come to know or learn about it once you start learning about the metabolism of the amino acid of the proteins. So the pyruvate, whatever happens as an end product of the glycolysis, it can be converted into acetyl-CoA, lactate, alanine, or oxaloacetate. These are the amino acids depending upon needs of the cell. So first one is the conversion into the lactate in the muscle and RBC. So this conversion of lactate from the pyruvate, it takes place in the absence of the oxygen. So pyruvate will be reduced to lactate and certain tissues, cell types, retina, erythrocytes, they convert the glucose into the lactose even in the aerobic conditions. So this is the one wherein the fate of the pyruvate will be there, that the pyruvate it gets converted into the lactate. That is the one thing. So this is the reaction formed in the presence of NADH converted into the NAD, the presence of the enzyme lactate dehydrogenase, pyruvate is getting converted into the lactate. This is the one fate that happens to the pyruvate. Second one is the conversion of alanine into the alanine or oxaloacetate. This oxidation of the pyruvate, it is very important in the process of catabolic reactions, and the pyruvate has the anabolic fates also. Not only the catabolic, but it is also has the results in the anabolic one. In this one, it can provide the carbon skeletons for the synthesis of certain amino acids like alanine or oxaloacetate. So you will come across this one clinically when you will be knowing about the metabolism of the proteins. That time the two reactions or the two tests are there based on the name of these two amino acids, alanine and oxaloacetate. That is we call as the alanine transaminoferase and oxaloacetate. That is we call as the AST. oxaloacetate transaminase. So these are the two tests we will do which are pertaining to inform us about the functioning of the liver, particularly the ALT we call that one, that is related to alanine, and oxaloacetate, that is, in other words, it is called as the aspartate one. So that one, it indicates about the liver and also the heart. So conversion of pyruvate to the acetyl CoA is the one which takes place very prominently, because once pyruvate is formed, it has to reach to the acetyl CoA, where from wherein it will be joining to the Krebs cycle or citric acid cycle. So it is converted to acetyl CoA by the oxidative decarboxylation, and it only occurs in the mitochondria. Remember well, the glycolysis takes place in the cytosol, whereas the Krebs cycle, it is taking in the mitochondria. So high activities regarding this PDH, I will take in the next class. High activities of PDS are found in the cardiac muscle and the kidney, and irreversible reaction shown by this PDH, it develops a link between glycolysis and the citric acid cycle. So here you can see that what will be the fate of this pyruvate were formed into the acetyl, converting it into the acetyl CoA in presence of pyruvate dehydrogenase. Now coming to the importance of the glycolysis. What is the importance? First thing is that it is the first major pathway which has been used for the production of the ATP molecules. It is a principal route for the glucose metabolism, and it provides the ATP in the absence of the oxygen and allows the tissues to survive in this anoxic episodes. So in the erythrocytes, glycolysis, it supplies the 2,3-BPC, which is required for the transport of oxygen by the hemoglobin. One is the production of the ATP, and by the help of ATP, it will help for the cells to survive. Third thing is that production of the 2,3-BPC, which is helping to transport the oxygen by the hemoglobin, even in the conditions wherein the person will be at the high altitude or in the anemic conditions. The fourth one is that it generates precursors for the other biosynthetic pathways, like the pyruvate is transferred to amino acid alanine for the, it acts as the precursor, and also for the synthesis of the fatty acids, and for the glycerol 3-phosphate, it is required for the synthesis of trialkyl glycerol, that is, which is you are finding that it is a product at the third step, where there will be the formation of glycerol 3-phosphate, which is required for the synthesis of triglyceride or trialkyl glycerol. So these are the things which will be made available from the glycolysis. From the glycolysis, you are getting ATP, availability of the ATP to the cells. Third one is the availability of oxygen by the HB, and fourth one, it is providing the 2,3-BPG. Fourth one is it is forming the precursor for the synthesis of some amino acids, and also it provides glycerol 3-phosphate for the synthesis of the trialkyl glycerol for the of the fatty acid. So these are the biological or biomedical importance of the glycolysis. Now we'll just start today with the citric acid. Citric acid is the another major pathway in the metabolism of the carbohydrates. It is also referred as the Krebs cycle or also called as the tricarboxylic acid cycle. So all the three names pertain to only one mechanism or the pathway, that is citric acid cycle. So it is the most important metabolic pathway to provide the energy to the body, and about 60 to 70% of the ATP is produced by this cycle, and it can be considered as a central hub for the metabolism since it acts as a connecting point of different metabolic pathways. Just to assume that a circle is there in the city from wherein the various other different roads they come and join there. So that way the citric acid cycle or the Krebs cycle is the one like a circle wherein different pathways they come and join together. It accounts for the majority of the carbohydrate metabolism and amino acid oxidation. This is the importance of the citric acid cycle or Krebs cycle. So what is the definition for that one? It is a series of reactions in mitochondria that brings about catabolism of acetyl CoA to form carbon dioxide and water molecule with the generation of ATP. This is the definition for the citric acid cycle. These reactions releases stored energy through the oxidation of acetyl CoA derived from carbohydrate, fat and proteins into the carbon dioxide chemical energy and in the form of the ATP. The end cycle that consumes acetate in the form of acetyl CoA and water, and what importantly they do that it reduces the NAD to NADH and produces the carbon dioxide, which we can consider as the waste byproduct. So NADH generated in the cyclic cycle is fed into the oxidative phosphorylation electron transport pathway. So that we will come to know more when we learn about the ETC. The net result of these two closely linked pathways is the oxidation of the nutrients to produce usable chemical energy in the form of the ATP. That is the form of the glycolysis and the Krebs cycle. The main important aim of these reactions are that oxidation of the nutrients and then releasing the energy in the form of the ATP. So it occurs in the mitochondria in contrast to the glycolysis which takes place in the cytosol. So all these reactions related to the citric acid, it was studied in the 1937 by the one scientist called Hans Adolf Krebs, and it is named by his name only, that is Krebs cycle. It is the one which is consisting of the 8 reactions. This is how it starts. So just we'll touch to the reactions of the TCS cycle. The major chunk of the acetyl CoA is produced from the pyruvate, which is in turn derived from the carbohydrates. You all know that the pyruvate will be formed from the glucose, and then pyruvate is converted into acetyl CoA, and further the other amount of this pyruvate CoA, it is obtained from the other things like the fatty acids and some amino acids. So just I told that one, as it acts as a herb, we can see that it is the one wherein the end products of other metabolism also get involved in this Krebs cycle. So as I said earlier, citric acid is the one pathway which consists of, or it occurs in the 8 steps. The first step is the synthesis of citrate. First step is the synthesis of the citrate. It begins with the condensation of 4 carbon oxaloacetate and 2 carbons ester COA. In this one, the enzyme citrate synthase, it contains the ester moiety of ester COA to oxaloacetate, forming citrate ester COA will be liberated. You can see here that from the oxaloacetic acid, which is having the 4 carbon one, it will be converted into the ester citric acid, where there will be the 5 molecules of carbons will be formed. So this is the first reaction wherein it takes place from citric acid, condensation of the oxaloacetate and forming the 2 carbon ester COA. So these are the reactions. Totally about 8 steps are there, which we will learn in the next class. Today we are seeing that one, mainly the glycolysis, which is having the 10 steps. Out of that, 3 are the one which can be considered as the irreversible one. Others are the reversible one. Irreversible reactions, the main intention of this is that to have the regularization. So intermittently with this one, the body can regulate or control the formation of the ATP molecules. So these are occurring with the help of 3 things. One is hexokinase or glucokinase. Second one is the phosphoglucokinase. Third one is the pyruvate kinase. Apart from this one, the regularization will be controlled by the hormones like insulin and glucagon also. Okay. So we will meet in the next class. Thank you.